Okay, today we're going to be working on the DNA Replication Lab. So Mrs. Wendt and I are here together, and Mrs. Baker-Merrigan. And we have our lab sheet that you're going to need. So go into Schoology and make sure you get that. And then we have assembled all of our materials. So all of our nitrogen bases that we're going to need in order to make our DNA. So we have A, T, C, and G. We have our original strand of DNA so that we know what we're making. So our instructions, and then we have our DNA replication map that we'll get to in just a little bit.
All right, so we are finishing up making our original stranded DNA here. Notice that all of the red letter A's are matching up with the the red letter A's are matching up with the yellow letter T's. And the green letter G's are matching up with the blue C's all the way across. And once we get this built, we're going to take the mat away. We'll be able to use the mat at the end of the lab to see if we made any mistakes. So one final piece here.
Oops, this one's going wrong. These are going the wrong way.
Oh. So you can see we made a mistake. We got to flip one of our pieces around. No big whoop. Okay, so we've got our stranded DNA here, and we are going to feed it through this orange guy here. And this is an enzyme called helicase. So like the helix shape on a DNA. And this is going to untwist and unzip the DNA. So you can see it kind of feeds through on one end, and the enzyme is going to break some of those hydrogen bonds in the middle, because those are, kind of weak bonds, easy to pull apart. And you can see that it just unzips super nice. Okay, now that some of our DNA has gone through helicase, we have a new enzyme here, these gray DNA polymerase. And they are going to build. Remember, poly means many, so we're going to build it up bigger. And you can see as both strands go through, the polymerase can work and add complementary bases to the other side. So they're not identical bases, but they're the match. So A's pair up with T's, C's pair up with G's. And you can see that as it gets fed through, we just keep adding one matching base at a time. Now in real life, your cells are going to do this extremely fast, but we're old. We do it as fast as we can handle. Okay, so the one that Miss Mills is building right now, this is called the leading strand, and it slips through the polymerase really easy. And you can see that coming out the end, we have, once again, a double or two strand of DNA. And we're going to check in a little bit to see if it's exactly the same as the strand we started with. All right, now the other strand of DNA here this is called the laying strand. And you notice here as we build, because of the shape of the enzyme, things just don't flow out really nice. So after you build a chunk, it kind of has to pick up and then go to the next chunk. So instead of flowing through evenly, this side called the lagging side gets built in a little chunk, and then it does the next chunk and does the next chunk. It still works effectively, but just because of the shape of how all the chemicals are bonded together, that's the most effective way to build this side. So we had the leading strand at the top, which flows through the enzyme really easy. And then down here is the lagging strand, which we have to build just one little chunk at a time. And if you notice, I'm going to have Miss Mills move her hands a second. Do you notice that the top and bottom strand that we've built so far, they're identical? Blue, green, down here, blue, green. So far, replication seems to be working. The two strands that we're building are exactly the same as each other. And the goal is that they will be exactly the same as the original strand we started with. Okay, so we are going to keep building here. The rest of the DNA will get fed through our helicase enzyme to be unzipped. And then our polymerase enzymes will do the building. And we'll come back and check and see if we had any mistakes where it didn't get copied correctly. All right, so we have finished building. So our enzymes now are free to go and build somewhere else. They are reusable over and over again. And we are left with these 2 strands of DNA. And if you look, both strands exactly match the original pattern we started with. So we have no mistakes, and those are called mutations. Now, when your body's building this, remember, they're doing millions and millions of these guys. So there will be occasionally a mistake, but you have some things in your body that proofreads and catches those usually. But sometimes mistakes go through, and sometimes there could actually be a benefit. Sometimes the mistakes can be deadly or be something not good. And sometimes the mistakes just don't matter at all. Okay, but this is DNA replication. We started with 1 double-stranded DNA, and we have 2 double strands that are exactly the same when we are all done. The enzymes we used for building can be reused to go replicate something else.